So good afternoon everyone, how are you all? I hope each and every one of you is in the pink of health and happiness. Yes, shall we start the class? Good afternoon. So today we are going to learn about biotechnology and its application. Okay, as usual we are going through the PYQs. Along with that we are learning the concepts. Okay, we have around 42 PYQs. It's going to be a short class. Okay, short duration. So our first question is from biotechnological application in agriculture. Okay, the BT toxin in genetically engineered BT cotton kills the pest by, how does it kills the pest?
Pores in midgut.
Create the pores in midgut. Very good. So this is the correct answer. It creates the pore in midgut. A is the correct answer. Following are the steps involved in action of toxin in BT cotton. Yes, the inactive toxin converted. Okay, we need to find the one second. Yes, we need to find the order of the steps, sequence of the steps. So just tell me, read the statements and tell me the sequence of the steps. It is the inactive toxin converted into active. Okay, the bacillus produce crystal with toxic inside the protein. Yes, the alkaline pH solubilize the crystals. The activated toxin binds to the surface of the midgut cells, create pores and cause death of the insect. The toxin proteins exist as inactive toxins in bacteria. Yes. The Bacillus thuringiensis produce crystals with toxic insecticidal proteins. The toxin proteins exist as inactive forms, that is protoxin in bacteria. The inactive toxin converted into active form due to the alkaline pH of the midgut. The alkaline pH solubilizes the crystal. Okay. The alkaline pH solubilizes the crystal first. Okay, the inactive toxin converted into active form due to the alkaline pH of the midgut. And the activated toxin binds to the surface of the midgut cells, creates pores and causes death of the insect. So it is BCEAD. BCEAD. Oh, BCAED. C. Okay, BECAD. BECAD. Yes, BECAD. Yes, correct. BECAD. Understood? Yes. So our next question. Given below are two statements. Statement 1, RNA interference takes place in all eukaryotic organism as a method of cellular defense. Is it correct?
Yes.
Yes, it is correct. And RNA interference involves the silencing of a specific mRNA due to the complementary single-stranded RNA molecule that binds and prevents translation of mRNA. Tell me. Is the statement 2 correct? Is the statement 2 correct? See, RNA interference involves in silencing of specific mRNA due to complementary double-stranded RNA, complementary double-stranded RNA, not single-stranded. So this is wrong. That binds and prevents the translation of mRNA. Okay, everything is correct, but single-stranded RNA, it is not single-stranded RNA, it is double-stranded RNA. So which makes this statement as wrong. So 1 is correct, 2 is wrong, 1 is true, 2 is false. So this A is the correct answer. Understanding? The capacity to generate a whole plant from any cell of the plant is called totipotency. With this totipotency nature, we can do tissue culture. Okay. Given below 2 statements, BD toxin are insect group specific and coded by gene Cry1AC. Yes. The BD toxin exists as inactive protoxin in Bacillus thuringiensis. However, after ingestion by the insect, the inactive protoxin gets converted into active form due to the acidic pH of the insect gut. Is it acidic pH?
No.
This is alkaline pH, not the acidic pH. So which makes statement 2 as wrong statement. So 1 is true, 2 is false. Again, option A as a correct answer. Which of the following are fused in somatic hybridization involving 2 varieties of plants? In somatic hybridization, which two of them are fused? Is it protoplast, pollen, callus, or somatic embryos? Actually, it is protoplast. Protoplast of the 2 varieties are fused in somatic hybridization. Which of the following statement is incorrect about Agrobacterium tumefaciens? It is used to deliver gene of interest in both prokaryotic as well as eukaryotic host. We need to find the incorrect. Tell me. TI plasmid from Agrobacterium tumefaciens used for gene transfer is not pathogenic to plant cell. The TI plasmid used in the genetic transfer is not pathogenic to the plant cell, correct? It transforms normal plant cell into tumor cell. Agrobacterium tumefaciens does that. It delivers tDNA into the plant cell. This is also true. But it used to deliver the gene of interest in both prokaryotes and eukaryotes? No, it doesn't.
No.
So we got it in the first option itself. That is, A is the statement A. This one is a wrong statement. So A is the correct answer. Transposomes can be used during which of the following? Is it during the gene sequencing, polymerase chain reaction, gene silencing, autoradiography? NCERT based question. In NCERT about transposome, retrotransposomes, they have written retrotransposomes. Which one? Where? Yes, in RNA interference. Yes, very good Kiran, very good Sakshi. In RNA interference, you can see that the transposomes are used in the RNA interference. That is gene silencing, it is used. So, C is the correct answer. Beta cotton variety that was developed by the introduction of toxin gene of Bacillus thuringiensis is resistant to fungal disease, plant nematodes, insect predators, insect pest. Insect pest. So 9, D is the correct answer. D for Delhi is the correct answer. RNA interference is used for which of the following purpose in the field of biotechnology? To develop pest resistant plant against infestation by nematode? Yes. To enhance mineral usage of plant? No. To reduce post harvest losses? No. To develop the plant tolerant to abiotic stresses? Wrong. So RNA interference is to make the plant resistant to infestation by nematodes. To this. So we got the answer, that is this one. A is the correct answer. Which of the following is true for golden rice? Which of the following is true for golden rice? It is vitamin A enriched with a gene from daffodil. Yes, this is the correct answer. Okay, A is the correct answer. Very good. What triggers activation of protoxin to active Bt toxin of Bacillus thuringiensis in bollworm? Easy.
Alkaline pH of gut.
Yes, yes, easy. That is alkaline pH of gut. The gut of the bollworms have alkaline pH where the protoxin will be converted into active Bt toxin. Which part of the tobacco plant is infected by Melargine incognita?
Root.
Root. There is a diagram also, figure also in the NCERT textbook about how there is a nodule formation taking place in the root due to the nematode infestation. In Bt cotton, the Bt toxin present in plant tissue as protoxin is converted into active toxin is due to, again repeated, third time repeating, yes. It is due to the alkaline pH of the insect gut. The golden rice is genetically modified crop plant where the incorporated gene meant for biosynthesis of
Vitamin A.
vitamin A. C is the correct answer. Which of the following Bt crop is being grown in India by the farmers? Is it soybean, maize, cotton and or Bt brinjal?
Cotton.
Mainly in India, cotton, Bt cotton is grown, Bt cotton. Biotechnology and application in medicine. Now you need to match 1 and 2, list 1 and 2. Genetically engineered human insulin
E. coli.
by the E. coli. GM, that is GM cotton.
Bacillus thuringiensis.
ADA deficiency.
Gene therapy.
ELISA.
Antigen antibody interaction.
Yes. So D3. C1. D4 Delhi is the correct answer. Which of the following techniques does not serve the purpose of early diagnosis of a disease for its early treatment? Is it ELISA, RDNA technology, serum and urine analysis or PCR?
Serum and urine analysis.
Directly from the NCERT, take this one, lines. Easy, yes. It is serum and urine analysis. Select incorrect statement regarding the chemical structure of insulin. Mature insulin molecule consists of 3 polypeptide chain ABC.
No.
No, they only contain A and B. So we need to find the incorrect statement anyway. We got the incorrect statement in the first statement itself. Match list 1 and 2. Gene therapy. Gene therapy is for allows the correction of yes gene defect. RNA interference.
Cellular defense.
Cellular defense in eukaryotes. ELISA.
Diagnostic test for AIDS.
Diagnostic test for AIDS. And gel electrophoresis. Separation of DNA fragments. So D1. C2.
Option C.
Option C is the correct answer. Again, yes, gene gun. Match 1 and 2.
Used to transfer of gene.
Used for transfer of gene. Gene therapy.
Replacement of faulty gene.
And gene cloning. To obtain the identical copies of a particular DNA molecule. Genome. Total DNA in a cell of an organism. Yes. A2, B1, C4, D3. B4 Bombay is the correct answer. Yes. Now, in gene therapy of adenosine deaminase, ADA deficiency, the patient requires periodic infusion genetically engineered lymphocytes because genetically engineered lymphocytes are not immortal cells. True. We need to find the correct answer. Yes. So, in the first itself we got the correct answer. Genetically engineered lymphocytes. Why they need periodic infusion of genetically engineered lymphocytes? Because genetically engineered lymphocytes are not immortal cells. So, they need to periodically infuse. Statement related to human insulin are given below. Which statement is or correct about genetically engineered insulin? We need to find the correct statement. We need to find the correct statement for genetically engineered insulin, which means humulin. For humulin, okay. Prohormone insulin contain extra stretch of C peptide. Correct. Prohormone insulin contain extra stretch of C peptide.
Yes.
I mean, as a statement it is correct. Yes, prohormone insulin has extra stretch of C peptide. But for what we are finding the correct statements? For humulin, that is genetically engineered insulin. Genetically engineered insulin में जैसे prohormone होता ही नहीं है ना. We are actually taking two different E. coli, and in one we are preparing A, in another we are preparing B, and A B के बीच में we are making the disulfide bridges, isn't it? So, do we have prohormone, like are we in the genetically engineered insulin, is there any C peptide? In genetically engineered insulin there is no C peptide. Okay, as a statement A, as a statement correct, but with respect to genetically engineering insulin, that is wrong. Okay, A peptide and B peptide chains of insulin were produced separately in E. coli, extracted and combined by creating disulfide bond between them. Correct?
Yes.
Yes, this is correct. Insulin used for treating diabetes was extracted from cattle and pigs. Yes. As a statement, insulin used for treating diabetes, like way back then, before genetic engineering, RDNA technology, were extracted from cattle and pigs. As a statement it is correct, but with respect to genetically engineering insulin, that is humulin, wrong. It has got nothing to do with that. Prohormone insulin needs to be processed for converting into mature and functional hormone. Prohormone insulin needs to be processed and converting into mature and functional hormone. This is in the normal, like normal human, like who are not diabetic. In them, first pancreas will be producing prohormone insulin, okay, where it has A, B and C chain, peptide chain. Later then what happens? That is the inactive form. Mature insulin, the C peptide will be out and only A and B will be joined together with a disulfide bond. That is in the normal human being. Now, genetically engineered insulin, this has got nothing to do with that. Some patients develop allergic reaction to the foreign insulin. As a statement, this was true, okay, because the insulin was from cattle and pigs. But with respect to genetically engineering insulin, this is also wrong. So, only B is correct. Understanding? This is a really good question, 2022 PYQ. Really good question, because we need to read the question very, very carefully, because it's said, because all the statements are themselves true. Okay, all the statements are true, but the question asked here is which statement is or true about genetically engineered insulin. They've only asked about genetically engineered insulin के लिए कौन सा और 5 से कौन सा ट्रू है, 5 में से. Understanding? Understanding? It's really good question. Really good question. Yes. When gene targeting involving gene amplification is attempted in an individual's tissue to treat disease, it is known as, this is known as,
Molecular diagnosis.
when a gene molecular diagnosis, when a gene targeting involves gene amplification is attempted in an individual tissue to treat disease, it is known as gene therapy. Here, because gene is, when the gene targeting, the gene is involved. Sometimes if there is a defect in gene, example ADA deficiency, in that particular gene is defect gene. So we are actually doing the gene therapy for it, and we are targeting that particular gene involving the gene amplification is attempted in an individual tissue to treat the disease is known as gene therapy. Which of the following is not an application of PCR? Gene amplification, purification and isolation, purification of isolated protein, detection of gene mutation, molecular diagnosis. Yes. So we need to find which is not, that is purification of isolated protein is not it's not an application of PCR. Understanding? Which of the following is a correct sequence of steps in a PCR? Correct sequence of steps. This is a repeated question, I think. Yesterday also we did this. Denaturation, annealing and extension. Easy question. Yes, very good, Kiran. D for Delhi is the correct answer. Nowadays, it is possible to detect the mutated gene causing cancer by allowing a radioactive probe to hybridize its complementary DNA in a clone of cells, followed by its detection using autoradiography, because mutated gene completely and clearly appears on the photographic film. No, mutated gene does not appear. Mutated gene does not appear on the photographic film. Yes, as the probe has no complementarity with it. Is the probe doesn't have the complementarity with it. That's why the mutated gene does not appear on the photographic film, as the probe doesn't have the complementarity with it. This is true. Mutated gene does not appear on the photographic film as the probe has complementary. No, it doesn't have complementary. So mutated gene partially appears. No, this is the correct answer. Before Bombay is the correct answer. Mutated gene does not appear on the photographic film, as the it doesn't appear on the photographic film because as there is no the probe is tagged with the radioactivity. Okay, the probe is tagged hota hai. Okay, if there is a photographic film, if the mutated gene is, the normal gene is there, then this probe goes and attached to it. Okay, this probe goes and attached to it like this. It is radioactive. So when you see this photographic film under the autoradiography, it can be clearly visible. This will be visible. This will be visible. Okay. For example, the gene, if the gene is mutated, for example, if the gene is mutated, this particular probe, the probe doesn't have the complementarity to go and bind to it. So the probe doesn't go and bind. If the probe is not there, the probe is the one which is radioactive. If you see under the autoradiography, this particular thing will not be appearing in the photographic film. Understanding? With regard to insulin, choose the correct option. C peptide is not present in the mature insulin. C peptide is not present in the just with regard to insulin they have asked. C peptide is not present in the mature insulin. Correct. The insulin produced by RDNA technology has C peptide. No. You can delete this too. The proinsulin has C peptide. This is true. A peptide and B peptide of insulin are interconnected by disulfide bridges. This is also true. A, C, D. A, C, D are true. B is the correct answer. Understanding? Yes. Very good, Kiran. For effective treatment of the disease, early diagnosis and understanding its pathophysiology is very important. Which of the following molecular diagnostic techniques is very useful for early detection? Tell me. For early detection, it is ELISA, PCR. Yes, very good. B is the correct answer, ELISA. The adenosine deaminase deficiency result in Parkinson's, digestive disorder, Addison's, dysfunction of immune system. Very good. D for Delhi, that is dysfunction of immune system. Which of the following statement is not correct? We need to find the incorrect statement. The proinsulin has an extra peptide called C peptide. True. The functional insulin has A and B chains linked together by hydrogen bonds. It is disulfide bonds. So we got the wrong answer, that is incorrect one, that is B for Bombay is a incorrect option. Incorrect statement, so B for Bombay is the right answer. Now match 1 and 2. BT cotton, BT stands for Bacillus thuringiensis. Adenosine deaminase deficiency, it can be cured by gene therapy. RNA interference, it is a cellular defense. PCR, detection of HIV infection by amplification. So 4-3. D for Delhi is the correct answer. Yes? Understanding? Which of the following is commonly used as vector for introducing a DNA fragment in human lymphocytes?
Retrovirus.
Retrovirus, yes. Very good. Which kind of therapy was given in 1990 to a 4-year-old girl with adenosine deaminase deficiency? It was?
Gene therapy.
It was gene therapy. The two polypeptides of human insulin are linked together by easy disulfide bridges. ADA is an enzyme which is deficient in a genetic disorder SCID. What is the full form ADA? Adenosine deaminase. Yes. The first human hormone produced by recombinant DNA technology is
Insulin.
before Bombay, insulin. Now match alpha 1 antitrypsin for emphysema, cry1AB gene, cry1AB is for corn borer. Very good. cry1AC is for
Cotton bollworm.
enzyme replacement therapy,
it is.
ADA. So D2, D2, A3, C1. Yes, A is the correct answer. Milk of transgenic cow Rosie was nutritionally more balanced product for human babies than natural cow milk because it contained
Human alpha lactalbumin.
human alpha lactalbumin. Human alpha lactalbumin. Now coming to ethical issues. Given below are two statements. Statement 1, the Indian government has set up GEAC which will make decisions regarding the validity of GM research. Correct? Correct. Biopiracy is a term used to refer to the use of bioresources by native people. Is it by the native people? It is called, yes. So this is wrong. It is not by the native people, non-native people. That is called biopiracy. So 1 is true, 2 is false. A is the correct answer. The laws and rules to prevent unauthorized exploitation of bioresources are termed as the laws and biopatenting. D for Delhi is the correct answer. Biopatenting. In India, the organization responsible for assessing the safety of introducing genetically modified organism for public use is Genetic Engineering.
GAC.
Yes, GEAC. Appraisal Committee. That is GEAC. A new variety of rice was patented by a foreign company through such varieties. Those such varieties have been present in India for a long time. This is related to basmati. Use of bioresources by multinational companies and organizations without authorization from the concerned country and its people is called
Biopiracy.
That is called biopiracy. B for Bombay is the correct answer. Which body of the government of India regulates GM research and safety of introducing GM organism for public services?
Genetic engineering.
Yeah, GAC. Okay, this was the last question. Understood? Any doubts? Okay, thank you so much everybody.